Good day, everyone. I'm glad to present this recent publication for our team in iMeta. Our study's title was multiPrime, a reliable and efficient tool for targeted next-generation sequencing. Comparing with MGS, TNGS has become an increasingly important strategy for exploring the crucial roles of the microbiome due to its high speed, cost efficiency, and broad range. Critical to the success of TNGS is primer design. Many factors such as melting temperature, GC content, and secondary structure play pivotal roles in primers' efficient target annealing. Many software programs have been developed to assist in primer design, but few are tailored to it, especially for the challenge of designing primers for large and diverse targets. We introduced multiPrime, a novel tool developed for TNGS primer design. multiPrime is a user-friendly and one-step tool for designing TNGS primer sites. It integrates degenerate primer design theory with mismatch handling, resulting in improved accuracy and specificity in detecting broad-spectrum sequences. It outperformed the conventional programs in terms of runtime, primer number, and primer coverage. The versatility and potential of multiPrime is highlighted by its potential application in detecting kinds of sequences. In terms of the ability to design degenerate primers, we compared the performance of multiPrime's primer design module with that of DG-Prime, a widely used program for designing degenerate primers for bacterial species that's been shown to outperform other programs. We discovered that the primers designed by multiPrime with one mismatch tolerance had significantly higher coverage than those produced by DG-Prime, particularly when the entropy of the primer region was less than 3. This advantage was even more pronounced when comparing combinations of primer pairs. Furthermore, multiPrime primer design module outperformed DG-Prime in terms of primer design efficiency. The primer set was utilized to amplify PCR products from 30 clinical specimens and 3 negative control samples. Then the product was sequenced using a long-read sequencing platform. All viruses were accurately identified with the target amplicon, and no virus reads were detected in the negative controls, indicating the high specificity of the primer set. To further confirm the accuracy and the effectiveness of the primer set designed by multiPrime, we designed a larger set of primers targeting 30 viruses, ensuring that 3-end mismatches were avoided. Subsequently, we evaluated this larger primer set using a cohort of 80 clinical specimens, and the primer set yielded a sensitivity of 87%, a specificity of 94%, and an accuracy of 89%. To access the efficacy of the primer set in capturing diversity and abundance, we devised primers targeting 16 epidemic pathogens and constructed the outcomes with those of MGS using 3 samples. The results demonstrate that our primer set facilitated target detection at a level comparable to MGS. Notably, all designed targets were successfully identified, with their corresponding recounts significantly surpassing those obtained through MGS. These findings underscore the effectiveness of the multiPrime designed primer set in capturing both the diversity and the abundance of target sequences. Overall, multiPrime offers an innovative and efficient approach for one-step primer set designed for TNGS. MultiPrime yields a higher number of candidate primers with higher coverage, resulting in a more efficient primer set with fewer primers. Furthermore, multiPrime uses a loss function to evaluate the likelihood of dimer formation, resulting in a reduced probability of dimer formation within the primer set and facilitating expansion of the primer set as needed.